Welcome, Brookings Bio students. This is Mrs. Rydell, and in this next part of our slideshow, we're going to see what happens next. We talked about our first thing that DNA does, which is make a copy of itself, and how the structure of DNA allows it to do that. And the other thing that DNA has to do in cells is to take provide information for the rest of the cell for how to do a variety of activities. And so we're going to look at how information is transmitted from the DNA that's in the nucleus out to the rest of the cell to tell it what to do. And we're going to need to talk about another kind of RNA. In our first chapter on molecules, we looked at two kinds of nucleic acids, DNA and RNA. And we compared the two, we looked at their structure. So this should be a little review for you. If you think back to the molecule chapter, we talked about the other kind of RNA. This is also made of nucleotide subunits, phosphate, sugar, and a nitrogen base. But we're going to change the subunit a little bit in order to make RNA. Instead of deoxyribose, we're going to put in ribose for our sugar. And remember, RNA is single-stranded, not double-stranded like DNA. And RNA is also different because we're going to change this is one of the nitrogen bases that we use. Instead of thymine, when we make RNA, we're going to use uracil. Remember, we have the A's and the G's and the C's still, but no thymine in RNA. We're going to use the U. And we're going to use three different kinds of RNAs to help with this process. If you remember back in chapter 1 in our molecules, we compared RNA and DNA, the shape, the structure, and also the function. And we said DNA is the genetic code molecule. It's going to stay in the nucleus. But RNA is able to take the information from the DNA out to the cell and use it to do protein synthesis. And so we're going to look at how that works. And there's three kinds of RNA that are going to help with protein synthesis. The first kind is called ribosomal RNA, or rRNA for short, R for ribosomes. Ribosomal RNA combines with proteins to actually form the little ribosomal subunits. And remember when we did our cell parts chapter and we colored all our cell parts, those little spots that were attached to the endoplasmic reticulum to make it be rough, those were the ribosomes. And ribosomes are made out of ribosomal RNA and proteins. If you remember from our cell parts chapter, we talked about where ribosomes are made. Remember that dark spot in the nucleus that's making the ribosomes, the ribosomal RNA? That's the nucleolus. And ribosomal RNA is made in the nucleolus, inside the nucleus, and goes out to the cytoplasm to form the ribosomes that are going to do our protein synthesis. The second kind of RNA is called transfer RNA. And remember, when we're doing protein synthesis, we're building proteins out of little subunits called amino acids. And the job of the transfer RNA is to bring in the correct amino acid and put it in the right spot. And it does it by matching up a spot on the message that's telling what amino acid to put where. We're going to talk how that works in just a minute. The third kind of RNA is called messenger RNA, and that's the RNA that actually carries the code from the DNA out to the ribosomes and provides the information on how to build our protein. Notice now these three kinds of RNAs have very different shapes. The ribosomal RNA is going to fold up into a 3D shape that forms those little ribosome subunits. Those are those little dots that we saw attached to our ER, or in some cases free floating in our cytoplasm. Transfer RNA has a very weird twisted shape, almost like a paper clip that's been bent. And all of these along here are A's and G's and C's and U's. But in this case, we just really showed these three because they're the most important ones at the bottom here to help the tRNA get in position. At the very end is the amino acid. Remember we said transfer RNAs are going to carry the amino acids and put them into the right spot. Message is linear. It's very straightforward, just like reading the lines on a page when you're reading information out of a book or off your paper. And so these three different RNAs have different shapes because of the jobs that they do. Okay, so we're going to start in the nucleus because that's where the DNA is in a eukaryotic cell. And we're going to open our DNA just like we did before and use the information along on the strand to fill in a complementary base match. Only this time, instead of adding in a DNA nucleotide to make a copy, we're going to add in an RNA nucleotide to make a message. When we did DNA replication, the enzyme that we used was DNA polymerase. We're making a polymer by joining DNA nucleotides. In this case now, we're going to make an RNA polymer. So use some common sense and think about, what do you think the enzyme is called that's going to help the cell do this? When we did making DNA polymers, we used DNA polymerase. When we're making RNA polymers, we're going to use RNA polymerase. This enzyme is going to separate the strands and then fill in with RNA base matches that make a strand of message. Now we're going to make an RNA message off of our DNA template. But it's going to kind of work the same way that worked when we did our replication. We're using this template strand to fill in the matching bases. So how does polymerase know where the gene starts and where it stops? If you take a chromosome and unwind it and totally stretch out the DNA, what you find is one big long piece of DNA. A chromosome is a long piece of DNA that has hundreds of genes lined up along on the DNA. Like think of them like boxcars on a train. And so the RNA polymerase, if it wants to make a copy off of one little segment of DNA to make a message from one gene, it has to have some directions about where to start and where to stop. Where does one gene start, and then where does it end, and then where does the next gene start? So we have some regulatory sites that are part of that DNA that are in between the genes. And so the first place we're going to talk about is called the promoter. Promoters are places on the DNA that are kind of like flags. Think about if you are landing in a plane at an airport and the plane is coming in, it needs to know which runway to go to and which one, where am I going to land? And there's lights there that flash, and there's flags, and it's telling the airplane, this is the place, put it down here. And the promoter does that same thing for the cell. There are places on the DNA right next to the gene, at the beginning of every gene, that are called promoter regions. And the promoters tell polymerase, RNA polymerase, this is the beginning of the gene. Start reading here. So think about those P's. That can help you remember what promoters do. Promoters are the place where polymerase attaches to start reading the message. And it's going to follow along on the DNA and transcribe the message. It's called transcription. We're going to transcribe the message off of the DNA, and it's going to follow along on the gene until it reaches an end codon. There's also stop signals at the end of each gene to tell polymerase, this is the end, stop reading. We're going to watch a little movie about how this process works.
Instructions for making proteins are coded by segments of DNA called genes. But proteins are not made in the nucleus. Instead, a strand of ribonucleic acid, or RNA, must carry the instructions for making proteins out of the nucleus. This type of RNA is called messenger RNA. Both DNA and RNA are long chains of nucleotides. Each nucleotide contains a sugar, a phosphate group, and one of 4 bases. RNA is different from DNA in 3 important ways. First, RNA is a single strand. Second, RNA has a different type of sugar called ribose in its nucleotide. Third, RNA contains the base uracil instead of thymine. The other bases found in RNA are the same as in DNA. They are cytosine, guanine, and adenine. The manufacture of RNA from a DNA template is called transcription. Transcription begins when the 2 strands of DNA in a specific region unwind and separate. Only one strand of the DNA serves as a template for the new RNA. The enzyme RNA polymerase matches RNA nucleotides with the complementary ones on the template strand of DNA. Recall that RNA contains uracil instead of thymine. Therefore, uracil pairs with adenine on the DNA strand. Transcription continues. C is paired with G. A is paired with T on the DNA strand. And U is paired with A in RNA. As the nucleotides are assembled in the proper sequence, RNA polymerase moves along the DNA strand, assembling the messenger RNA, until the entire gene has been transcribed. Hydrogen bonds reform between the complementary bases in the DNA molecule, reattaching the 2 strands. The coded instructions on the DNA molecule have been copied to the newly manufactured strand of messenger RNA.
So let's go back to our slideshow now and take a look at what's happening here. We're going to open our strand of DNA. We're going to use one side, and different genes are transcribed off of different segments of DNA. Sometimes we use the one strand, sometimes the gene is on the other strand. It kind of depends on what gene we're using. And we're going to use our DNA now to make a message. We're going to put down RNA polymerase at the promoter. We're going to run along on the code. We're going to read the information in the DNA, and we're going to add in RNA bases now, RNA nucleotides, to make a message that comes off of the DNA, and it's going to take that information out to the cytoplasm and use that code that's in the message to make a protein. This is the directions for making a protein. What amino acid goes where? We talked a little bit about regulatory sequences on DNA, and leads us to the idea of non-coding DNA. Not all the DNA that is part of the genome actually codes for proteins, actually carries information that's going to be turned into a message. And majority, in fact, of human DNA, almost like 90% of human DNA, is actually what we call non-coding DNA. Originally, we kind of called this we call it junk DNA, and because scientists really thought it was kind of extra, it wasn't really important. The important part were the parts of the DNA that coded for proteins and carried information on building amino acid sequences. But since that time, we've kind of learned a lot more about this junk DNA and finding out more and more about the function, and we're seeing lots of important things are carried in code that is not going to actually end up in protein. First of all, we do have some regulatory sites. We talked about promoters. There's other sorts of regulatory sites that make molecules called repressors and other regulatory molecules. We're going to talk a little bit more about those in our next slideshow. But there are regulatory sites that turn genes on and off that are in addition to the actual part that codes for the protein. We also talked in our cell parts chapter in our mitosis chapter about telomeres, protective ends on the ends of chromosomes. And remember, these are non-coding regions that are added to the ends of the DNA sequences that are added that protect the tip. Remember, every time the cell copies the DNA, a little bit of the code is lost at the end. And so those protective ends, if we lose those rather than losing important code, that is going to protect the DNA information. So telomeres are important protective sequences on the ends of chromosomes, and that's part of that junk DNA that doesn't actually code for proteins. The other pieces are the introns. When we talked about editing introns in removing them from DNA, we're going to talk about that in a minute. There's pieces in the DNA code that don't actually end up in the final message. We have pseudogenes. Pseudo means false, or if you think about these are genes that were functional at one time, maybe in past ancestors, that mutations have happened to them to make them no longer function, but the gene code is still in the genome, and maybe related to past ancestor functions. Example would be smell receptor genes. If you think about smell receptors in some kinds of organisms have very strong ability to sense different kinds of smells compared to humans. Think drug dogs or dogs that hunt for lost people. Bloodhounds, for example, have the ability to smell things that humans can't smell. They have receptors in their nose that allows them to detect odors that we can't detect. If you look inside the human genome, we have those same receptor genes, the same receptor genes that are in those organisms that can smell and detect odors are in humans, only ours are broken. Ours have mutations that make them non-functional. So the gene code is still there, but the genes no longer function anymore. And we have lots of pseudogenes in our genome that show connections to past genes that functioned at one time in ancestor species. Some of the what we call junk DNA is left over from viral infections. When we talked about viruses infecting cells and taking over the host genome, using the cell machinery in the cell to make virus, one of the other things that viruses can do is to hide out inside cells, remain in the cell instead of bursting the cell, and kind of hitchhiking along in the genome. They actually incorporate their genes into the bacteria and or into the cell that they are infecting and hide out as symbionts. And if you think about viral infections, if you think about things like chickenpox, if you ever had chickenpox before, that virus is still hiding out in your body somewhere. It can lead to outbreaks of shingles later on when you're older. It's that same chickenpox virus that's been incorporated into the DNA of your cells and is hiding out in there. And so some of the junk DNA that's extra in our genome has to do with past viral infections, and those leftover viral elements that are in our cells may be mutated now so they can no longer jump out. They're evidence of past viral infections in past ancestors also. And the last bit of non-coding DNA has to do with DNA elements called transposons, and we're going to talk about transposons in just a second. So if we look at some of those sequences that don't end up in the final message, one of the examples we said were introns. And if you look at this as a piece of DNA, part of that code in the DNA is going to end up in the final message that gets sent to the ribosomes and built into a protein. The part that ends up in the message is called the exons. Exons are sequences in the DNA that are going to end up in our final message to our cell and going to be expressed in the cell. In between, though, there are some in-between sequences called introns. And introns are
copied off the DNA when we make the first message, but they are edited out. They're removed before the final message. It's kind of like when you write an English paper and before you turn in your final copy, you do some editing to get it into the right shape and fix it the way you want it. And so the DNA information is built into or copied off into an RNA. We call it a pre-RNA. And then it's edited. The introns are removed and the exons are spliced together to make one message. Why would a cell do that? Why would it spend energy making a message, and then editing and throwing pieces away? And the reason scientists think is related to the idea of using the same gene sequence, the same DNA code, in a variety of ways that allows the cell to adapt. For example, one of the molecules that your immune system makes to fight germs is called an antibody. Antibodies are proteins, sequences, and they have a portion of the antibody molecule that's the same in every antibody. But if you think about it, antibodies are directed against invaders, right? Every time you have a germ or a virus enter your body, it's a little bit different. Measles virus is different than mumps or AIDS, or think about bubonic plague or strep throat or any of the other diseases that you could get. Each of those germs or viruses have proteins that are recognized as this is an invader, and your body is going to make a specific antibody to fight that germ. So the antibody that fights measles is going to be a little bit different than the antibody that fights chickenpox or fights AIDS or fights any of the other germs that you can be exposed to. So the ends of the antibodies are variable, and they vary depending on the germ they're being made to fight against. And so it allows our antibody gene, the little piece of DNA code that tells the cell how to make antibodies, to vary the code, vary the information, depending on what kind of germ is being fought. And it allows that same DNA code to be used in a variety of ways. Each time you make the message, you can edit it to adapt to whatever germ the body is facing. The other type of junk DNA or non-coding sequence that we talked about on our list was transposons, sometimes called jumping genes. And jumping genes are segments of DNA that seem to have the ability to move around from one place to another. They can jump from place to place in the genome. They were first described by Barbara McClintock. She was doing research with corn and looking at colors in corn and the expression of color genes. And she reported that seems like there were sequences in the DNA that seemed to jump in and out of other genes to move around in the genome and cause differences in the color patterns that she was seeing. She reported her research, and everybody laughed at her. They said she was wrong, that she was crazy, that genes don't move, they stay in one place. That's kind of what was accepted at the time. And she kind of went back to her laboratory and just did her research for many, many years. And eventually she was vindicated. We did discover these jumping sequences, jumping genes or transposons in other kinds of organisms, including humans. And they do seem to be able to jump and move from place to place. And they can take little pieces of DNA with them when they jump. It can help in genetic recombination, increase the mutation rate. They seem to jump more frequently in situations when the organism is stressed. Think environmental stresses like heat, drought, cause the increase in these jumping genes, and maybe related to organisms being able to come up with new gene combinations to adapt to new situations. So this, what we call junk DNA, is not really junk at all. It allows the same gene to be used in a variety of ways in different situations in different cells. Same DNA edited in different ways. A lot of what's different and makes organisms different is not about the basic sequence of their genes, but it has more to do with those regulatory sites. If you look at DNA in chimps compared to humans, more than 98% of their DNA is identical to ours. And what's different about the chimp and human DNA is not the actual genes that code for proteins. If you look at hemoglobin in chimps and in humans, they have the exact same hemoglobin gene as we do. And so it's not that the individual genes that code for proteins are different, it's how those genes are regulated, how they're turned on, they're turned off. The differences are in those regulatory sequences. And junk DNA may play a role in evolution. Small changes in genes can have a big effect. And think about things like viral DNA, jumping genes, those transposons, allow the cells to change based on environmental changes that they encounter.
Okay, so here's how this works. The DNA is going to stay in the nucleus. That's our master plan. We're going to make a message off the DNA. That's called transcription. DNA to RNA message is transcription. We're going to process, we're going to edit that message a little bit. And this is all going to happen where the DNA is. That's in the nucleus. We're going to send that message out. How does it get out? Remember, our nucleus has nuclear pores to allow things in and out, little doorways. And we're going to send that message out of the nucleus out to the cytoplasm, and the ribosomes in the cytoplasm are going to translate that message, turn that RNA message into a protein. So think of DNA as kind of like the master plan. When an architect makes a plan for building a building, the master plan stays in the main office, and copies are made of the plan with the directions that are sent out to the job site as working copies. So the builders, the contractors have a copy of the information of what to do, but it's a working copy. So if coffee gets spilled on it, if it gets a little dirty, I still have that master plan saved back in the office. And that's how the RNA messages work. They come out, we use them. Once they're used, we can destroy them, and I still have that master plan in the nucleus. So let's look at how the cell is going to turn that RNA message into a protein. And remember, we only have 4 letters, U, C, G, and A. And in order to provide, use those letters to provide information, we have to take those letters and group them together to make words. The message that comes from the nucleus is read in little groups of 3 called a codon. Each little group of 3, UCG, is a codon. This is a codon. It's telling the code or the information. And each of these little codons stand for a different kind of amino acid. So UCG stands for an amino acid called serine. CAC stands for a different amino acid. It's a different code. GGU stands for a different amino acid, glycine. Each codon represents a different amino acid. And remember, there are 20 different amino acids that are used to make proteins. So we're going to use kind of a little decoder wheel here to help us figure out what the messages say. And so I'm going to show you how this works. There are 64 possible codon combinations. If I take those A's and C's and U's and G's and combine them in different ways, I can make 64 possible codes. And notice on the wheel now, some of these amino acids have more than one codon that stands for that amino acid. So we're going to practice using the code. And remember, we read in little groups of 3. So the first letter of the code, we start in the middle of the wheel. So I have 4 choices. I could have an A as my first letter, a G, a U, or a C. The second letter of the code is found in the second ring. And as we move toward the outside, we're going to read the code, the codon, and figure out what amino acid it stands for. The third amino acid in the code is the next outside ring. So let's do a couple together. For example, let's try UCA. If my code says UCA, I'm going to start in the center. Here's my U. C is my second letter. And A is my third letter. And that code, UCA, is going to tell the cell, put in the amino acid serine. What if my code was CCA? That's a different codon, right? C is my first letter, C is my second letter, and A is my third letter. It's going to tell me to put in a different amino acid. It tells me to put in proline. So we can read our code wheel now. You don't have to memorize all the codes and memorize what the amino acids are, but you should be able to use the code wheel like this. If I gave you a message codon, you should be able to read the wheel and tell what amino acid it stands for. Notice there are a couple codons that don't code for amino acids at all. Like we have stop codons here. The very first codon, starting codon, is always AUG. AUG. So let's look at that. If I start my protein with AUG every time, what amino acid is going to go in at the very beginning of every eukaryotic protein? AUG. Methionine. What that means is every protein is going to have methionine in its first starting amino acid. Those may be changed later on, but as the protein is being made, every protein starts with methionine. It's a connection between living things on the planet. All living things, it doesn't matter if you're a human, a chimpanzee, a tree, a bacteria, a fungus, they're all using those same 20 amino acids to make their proteins, and they're all using the same genetic code, they're talking the same language. And it's a connection. It shows that connection between living things. Notice we have 3 codons on our wheel also now that are what we call stop codons. They tell we're at the end of the message. And so we have 3, UAA, UAG, and UGA. That is the stop codon. So let's see how this works now. This is going to be my message, my messenger RNA that comes from the nucleus. We're taking that off the DNA now, and we're going to use our three kinds of RNA to help us put this, take the information in the message and turn it into a protein. The way we do that now, remember, proteins are made by joining amino acids. So the transfer RNAs are going to bring in the amino acids, and how they know where to go is they're going to match a little section on that tRNA with the codon in the message. So if my first, remember, AUG is our starting codon, AUG is going to put in methionine. Here's methionine sitting up at the top here. And AUG now, I want to make sure methionine goes right in that spot. So on the tRNA that's bringing in methionine, I need a match to make sure it's going to sit where it needs to go. So the match for A is U. Remember, RNA doesn't have any T's. My match for A is U. A matches with this U, and C matches with G. This place, this is called the anticodon. It's opposite the codon in the message. We're going to match up our anticodon with our codon. It brings in the amino acid and sets it right in that spot. And each kind of tRNA is going to carry only one kind of amino acid. That amino acid is special just for this codon, and so it's going to match the anticodon and this carrier molecule to make sure that amino acid gets put in the right position. So we're going to bring our information out from the nucleus. We're going to attach the ribosome to the message. We're going to bring in the tRNAs to bring in the amino acids and set them down in the right position. And then we're going to link these amino acids together to make the protein. It's kind of like an assembly line. So the amino acids come in, the tRNAs are going to put them into position. Once we hook them together, the empty tRNA leaves the ribosome, goes out and can get charged up again with a new amino acid and come in again farther down the line. And we're going to keep adding those amino acids, bringing the tRNAs, bringing them in, putting them in the right position, hooking them together until I get what's called a polypeptide. Peptide bonds are the kind of bonds that hold amino acids together. So polypeptide, many amino acids hooked together. This is are the beginning of our protein. And then once we get to the end of the line, we're going to keep adding amino acids. When we get to the end of the line, we're going to release our protein. So we're going to watch a little movie about how this whole process works.
A protein is a chain of amino acids. The process of linking amino acids together to make a protein is called protein synthesis. Protein synthesis occurs in a cell's cytoplasm. mRNA leaves the nucleus through a nuclear pore after it is transcribed from DNA. The assembly of protein in the cytoplasm takes place on ribosomes. Ribosomes consist of two subunits, a small ribosomal subunit and a large ribosomal subunit. Floating within the cytoplasm are amino acids and transfer RNAs, or tRNA. Each kind of amino acid binds to one specific type of tRNA. Notice the 3-base region on the tRNA. It is complementary to a 3-base region on the mRNA. Each group of 3 bases on the mRNA is called a codon. The complementary 3-base sequence on the tRNA is called the anticodon. A small ribosomal subunit binds to the mRNA at the start codon, AUG. A specific tRNA carrying the amino acid methionine binds to the mRNA. The anticodon on the tRNA matches the codon on the mRNA. The large ribosomal subunit now binds to the small ribosomal subunit and the mRNA. A second tRNA with its attached amino acid binds to the second codon of the mRNA. The first amino acid is transferred from its tRNA to the second amino acid, and a bond between the two amino acids is formed. The protein chain has begun to grow. The first tRNA detaches from the mRNA. The ribosome moves down the mRNA to the third codon. This codon is recognized by the anticodon of another tRNA carrying an amino acid. The growing protein chain is transferred to the third amino acid. The second tRNA detaches. The ribosome again moves along the mRNA. A codon is exposed. Again, the codon is matched by the anticodon of a tRNA carrying an amino acid. The protein chain is shifted, adding another amino acid to the chain. The tRNA that gave up its amino acid detaches, and the ribosome moves down the mRNA strand to the next codon. This process, called elongation, builds proteins one amino acid at a time. The 3 bases that make up a codon determine which tRNA attaches. Since each tRNA carries a specific amino acid, the sequence of amino acids in the new protein is determined by the sequence of bases in the mRNA. The ribosome continues to move along the mRNA. At the end of the mRNA, the ribosome encounters a stop codon, such as UGA. The completed protein is released. The tRNA floats off, and the mRNA detaches from the ribosome. The ribosome splits into small and large subunits.
So let's go back to our slideshow. And look at the things we have learned about what happens in the cell and what DNA can do. The first process that we looked at was using DNA to make a DNA copy, a replica. Using DNA to make DNA is called replication. The other thing that we did was use the DNA information to make an RNA message. Going from DNA to RNA is called transcription. We're transcribing the information into DNA, from DNA into RNA. We're taking the information in the DNA and transcribing it into RNA. The other thing we did then is to take that RNA message and turn it into a protein. The process of going from RNA to protein is called translation. We use that word translation when we talk about languages, right? Translating English into German or German into Spanish. And the reason we use translation to talk about making proteins is that we're actually switching our alphabet. Remember when we made DNA or we made RNA, we were using nucleotide language, right? We were building it out of nucleotide subunits. When I make proteins now, I'm switching my language. I'm switching to amino acid subunits, and that's why it's called translation. We're going from nucleotide language to protein language, amino acid language, and that's why we use the word translation. You should be familiar with those three words and what they mean. This kind of leads us to what we call the central dogma of biology. How is information passed in cells? Information starts in the DNA. That's our genetic code molecule. But we're going to pass that information to the RNA by making RNA message. And then the RNA message is going to be translated into a protein. And remember, the proteins are the workhorses of the cell. Proteins are molecules that do things. They are transporters. They're structural. They act as enzymes. All of those things we learn that proteins do. When the protein does something, that's going to show up as a trait, a phenotype in our organism. That's how genes express themselves, how you get blue eyes or brown hair or be a certain height. It's because the information in the DNA is made into an RNA message and turned into a protein that's going to do something in your body, in your cells, like make a blue eye color protein, or make an enzyme that acts as a growth hormone to make you tall, or make a pigment in your skin that gives you a certain skin color. DNA acts through proteins to give you your phenotype. Proteins are the connection between the gene code in the DNA and how that gene is expressed. So a gene that codes for an enzyme, remember enzymes are proteins that make a pigment, can change the color of the flower. A gene that codes for an enzyme that is adds carbs to the glycoproteins, that's what's going to make your blood type. Enzymes catalyze chemical reactions, and remember we talked about enzymes that don't work. Like if you have a mutation in that gene for something like PKU, you're going to get a different phenotype. We're going to stop right there and do a little act it out to see how this protein synthesis works. And thanks for watching.